Okay, so this will be lecture 3. Cytology should be about a 30-minute one, and this is PowerPoint B. PowerPoint B as in boy. Okay, so it's not a whole lot out of this chapter. This is chapter 2 in your book. We're just going to delve a little bit into microanatomy, the cytology, which is, has to do with cells. Remember, cytology versus histology. This is just, sometimes I put something like this on a quiz, and I say pick out the proper, which one's in the proper order. Just make sure it goes from smallest to largest. Chemicals make up cells, cells make up tissues, tissues can make up organs, organs make up organ systems, and then organ systems make up the whole organism. So like an organ like the stomach, it can have 4 different tissues in there, right? It can have epithelial, it can have muscle, stomach, and in the muscle in the stomach, smooth muscle. It can have neural, it can have connective tissue. So an organ can be composed of 2 or more tissues. And then an organ system, I want you to think like, the respiratory system or the digestive system. There are several components of those. Okay. So back to cytology. Cytology is the study of the structure and function of cells. Cells are the structural building blocks of all life, the smallest structural unit that performs all vital functions. 2 main types, and that would be reproductive and somatic. So reproductive, sperm and egg. That's it, and that's that. The human body is made of about 75 trillion cells. I've heard 100 in some cases, 100 trillion cells. Most of those cells are what you call somatic cells. So somatic cells are all the cells of the body, like muscle, bones, fat, neural, skin, blood, immune cells, everything that composes those. That's somatic cells. Reproductive, simply sperm and egg. Okay, this is just showing you differences in what a cell can look like. Look at a neuron compared to this is cardiac muscle. You know it's cardiac muscle because you see you have these beautiful intercalated discs right here and right there. You remember cardiac muscle is branched. It's singly nucleated for the most part. Every now and then you'll find 2, according to some of my buddies. It has intercalated discs. There's little stripes, although you can't see them very well right here. Those are called striations, just like skeletal muscle has striations. And they're running in the same direction as these intercalated discs, but you just can't see them very well. So cardiac muscle is involuntary. It's singly nucleated. It's striated. Okay. So it's involuntary, not under your control. Oocyte, just showing you how big an egg is compared to a sperm. That's showing you a spindle-shaped smooth muscle cell. It also has 1 nucleus, singly nucleated. There's an osteocyte, bone cell, blood cells, fat cells, neurons, just showing you the difference. Here's some epithelial cells, just showing you the difference in morphology or the outward appearance of these cells, depending on which genes are turned on or off during their, development. Okay. Plasma membrane, which is a phospholipid bilayer. It's the cell membrane. They also call it the plasma lemma sometimes. So it isolates the cell from the environment. It's a semi-permeable membrane. It regulates interactions with the environment. Some things can go through it, some things can't. So you'll have intracellular fluid inside the cell, extracellular fluid outside the cell. So the membrane selectively allows the passage of water, nutrients, gases, waste, secretory products, ions, and gases into and out of the cell. So some things can go across those membranes, some things can't. And remember size, shape, charge, and solubility. That's the 4 characteristics of a molecule that can determine how easily or if a molecule can pass through that membrane. So the structure of the plasma membrane allows for its selectivity. And so you have hydrophilic heads on these phospholipids, and they're amphipathic, which means the end of this molecule, it has two ends, the head and the tails. So the ends have different affinities for water. So the hydrophilic head, it loves the water. It doesn't mind being in it. The hydrophobic tails, they do not like the water, so they want to be out of it. So this will orient itself, the membrane. When you get a bunch of these molecules together, all the heads will be lined up on the extracellular surface and on towards the inside, towards the cell. It will be heads, where in between those two heads will be tails. So it'll look like this. So you got heads pointing to the outside, heads pointing to the inside, and then the tails are in between them. So these are hydrophobic. These do not like the water. They're trying to get away from them. So you can see the water here and the water here. The heads like it, so they're facing towards it. And it just naturally takes on that configuration. So hydrophilic loves the water, hydrophobic hates the water. Phospholipid bilayer has two layers arranged with the hydrophobic regions facing each other, so that would be the tails, these fatty acid tails. Okay, now here's the tails, and notice you got some cholesterol scattered in between, and so that cholesterol is very important. How much or how little you have influences the rigidity or the fluidity of that membrane. So things float around in this membrane. It's like I said, I think I told you guys, I think they did a video called The Inner Life of Cells, Harvard. See if you can Google that. It should come up. And the artist did a little video of what they think the phospholipid bilayer would look like in real life if you could shrink yourself down, and the inner, mechanisms and organelles inside of a cell. They did those too, and they animated those. It's really, really nice. Has a good soundtrack to it too. So the proteins. What are proteins, first of all? So they're basically composed of amino acids. So amino acid hooked to an amino acid, hooked to an amino acid, hooked to an amino acid. You can have many, 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 many of these hooked together by these what they call peptide bonds in between them, which we're not going to go into. But these proteins can take on all different configurations, like three-dimensionally, and so they can become things. So like a channel or a receptor, right? Or they can become the framework of what's inside of your cell, with the filaments and microfilaments, things like that. So let's look at these. I'll go back, don't worry. So this is a channel. It's a bunch of protein. It's made of protein, and it's integrated into the membrane. So it's an integral protein. It just happens to be a channel. Here's a protein that's just on one side of the membrane. So they call that a peripheral protein. Here's one that's integrated, but it has like a carbohydrate hook to it, which, glyco, think sugar. So this would be an integral glycoprotein. Here's your cholesterol scattered around. Yeah, you got glycolipids, but they're not showing that too well. Here's some of that framework I was telling you about, the microfilaments, right? So there's a lot of structure inside of a cell that they didn't realize was there until they got the electron microscopes and they could see them. So proteins, like I said, they're made of amino acids hooked together, right? Carbohydrates, mainly think sugar in this case, on the outer surface of the membrane that can help function as a receptor. You can have glycolipids and glycoproteins. We showed you those. There's the cholesterol that's getting in between the tails, and it's actually kind of breaking it up and making it where it's not packed so tightly in this case. So anyway, you'll have cholesterol scattered throughout that membrane. And then also cholesterol is used for other things, like it can be the backbone for certain molecules or a hormone or whatever. Okay, so these were the four I was telling you about: size, shape, charge, and solubility. And solubility in this case, is it going to dissolve in water, or is it going to dissolve in lipids or fat? That's what they're talking about. So let's go back up here. Passive transport. This is not cell and molecular. So basically, that is, if something can go across the membrane, like down its concentration gradient, but it doesn't cost any energy, that's passive. No ATP is involved. Where active transport, you have to use energy to get that molecule across. Okay. So I could go, active transport uses ATP, true or false? And you go, true. Okay. Passive transport uses ATP. Just go false, even though, yeah, you got to spend energy to maintain that concentration gradient, but we're not counting that. Okay. Don't worry about filtration. Don't worry about facilitated diffusion. We're not going into those. This is anatomy. Just know a little bit about diffusion and osmosis. So diffusion, like we said, tendency for molecules to spread out from each other. So the molecules can move from a concentrated area to a less concentrated area. For instance, if you sprayed perfume at the front of the room, eventually the molecules would bounce around, and somebody at the back of the room would be able to smell it. So it diffused across that room from a high concentration of perfume to where it was low. Okay. The membrane, can something diffuse across the membrane? Yeah, if it's got the right size, shape, charge, or solubility, and a concentration gradient. The membrane, that would help. The membrane selectively restricts diffusion into and out of the cell. Okay. Whoop. Don't know what happened to my PowerPoint here. There we go. Okay. So large molecules may not be able to get across. If you have a charge on there, you may not be. Say you have sodium. Sodium has a charge on it, right? Positive charge. So it may not be able to fuse through that membrane, but you may have a channel that's specifically for sodium that can open up under certain conditions. And generally, you have more sodium outside of a cell membrane than you do inside of it. Intercellularly, right across the membrane I'm talking about. I'm not talking about way far away. Then that's a concentration gradient. You open that channel, the sodium would flow through. Potassium, generally you have more inside a cell membrane usually than you do outside. So if you open the potassium channel, then the potassium would flow out down its concentration gradient. Don't stress on that too much. I'm just showing you the way concentration gradients work. The diffusion of water. Basically, think of osmosis as diffusion of water. So think of the water as having a concentration gradient. So it can flow down its concentration gradient. So to hopefully make that make sense. Say you had a test tube, a U-shaped tube, and you had a semi-permeable membrane here. Now, don't pay attention to the way this is written right now, but let's just say you got a bunch of sodiums over here and not many sodiums over here. But the sodium, if it could get across this membrane, then it would just go down its concentration gradient until you had about the same amount of sodiums on each side. So nothing would happen to the water level because then you would be equal. Now, the way they drew this, let's act like the sodium cannot get across. Okay, so now we relatively have more water over here than we do over here per solute molecule. So the water can go down its concentration gradient, and that's an actual force. So when it does, look what's happened. The water level rose over here, until you equaled out and you have the same amount of water per solid molecule over here as you do over here. And that's an actual force. How does that come into play in anatomy and physiology? Well, just think if you had a red blood cell. Think of it as salty inside the red blood cell, although it's different than that. There's other molecules. And then you drop that red blood cell into a cup of pure water. What's going to happen? The water is got a higher concentration outside that cell than it does in it, so it's going to flow into the cell. And what's going to happen to that cell? It's going to swell up and possibly burst. So that's why if somebody comes into your emergency room and they're dehydrated, you cannot just give them an IV of water. It would probably kill them. It has to be osmotically balanced. You know, it's got to be the proper osmolarity. And you'll learn about all that in nursing school. It's a little too detailed for us to go into, but that's why this concept of osmolarity is important. Okay, so just the basics, this anatomy. Don't worry about facilitated diffusion. Don't worry about filtration. Active and passive transport, just know that active uses ATP, and for our purposes, we're saying passive does not. This, don't stress on it. We'll talk about this more when we get to neuro test 4, but it's a sodium-potassium pump. And you can see what's happening. Sodium is getting pumped outside of the cell, and potassium is being pumped inside of the cell. Remember what I told you, generally you have more sodiums outside and more potassiums inside. So in both of these cases, you're going against your concentration gradient. So you're pumping sodium out to where it's already high, and you're pumping potassium in to where it's already high. So that's going to cost you energy, and they're showing you ATP is being used. So, like I said, don't stress on that too much. I can go, true or false, sodium potassium pumps use ATP. True, right? Because they're pumping against the gradients of sodium and potassium. All right. Endocytosis, what does that mean? That just means going into a cell, basically. So moving molecules into the cell. There's 3 basic types: phagocytosis, pinocytosis, and receptor-mediated endocytosis. That's a mouthful. All right, let's take them one at a time. Phagocytize means to eat. So think of an amoeba, the way it has the little pseudopods that come out and they can wrap around a bacterium. Well, we have cells that can do the same thing, like macrophages. Macrophage, phagocytize, to eat, right? And so the macrophage can, it can surround the bacterium, bring it in, and then hopefully kill it off with these enzymes from the lysosome. Well, if it does, and you got to get rid of the stuff, that's exocytosis when it's going out. Generic word, just when you're putting stuff outside of a cell, it's exocytosis. Pinocytosis, that's another form of endocytosis. Kind of a cool one. Basically, it just pinches off and invaginates and pinches off, and whatever extracellular fluid is out here ends up in here. It's not specific, just whatever's out there comes in, and then you took it into the cell. So I think so it's like taking a drink, it's like the cell took a little drink of the extracellular fluid. So the way I remember it, like pina colada, you're taking a drink. I know it's spelled differently, but it reminds me of this. Right? It's kind of stupid, but it works for me. So pinocytosis, that's just taking a drink of the extracellular fluid. Receptor-mediated endocytosis, it's a selective form of pinocytosis. The vesicles contain a specific molecule in higher concentration once it invaginates and brings it in. Say there's something specific out there that you want. All right, I'll come back to that. Okay. Ligand or ligand, I've heard it pronounced both ways, generic for just whatever kind of molecule. Say this is a specific ligand and it's specific for this type of receptor. Once you get a bunch of them loaded on here, then that's a signal, and then this membrane will invaginate and bring that in. So what do? Yeah, you bring in some of the extracellular fluid, but you're bringing in a really high concentration of this particular ligand or ligand. So it's just a real specific form of pinocytosis. So the ligands or ligands bind to the receptors, the vesicle forms and brings in specific molecules. Okay, so you see how I could do a matching. Maybe I could go Exocytosis, penocytosis, receptor-mediated endocytosis, and then I could make a little description, and then you could match them up. Or I could do give me a little one-sentence blurb, describing each of these. I could do something like that. Or I could do a multiple choice. Okay. Exocytosis we talked about is just bringing a product out. Say this is a hormone or a neurotransmitter or whatever. That little vesicle can fuse with the membrane, and then look what it's doing. It's spilling out the molecules outside of the cell. Okay, and that's a little more complicated than they're making it out to be, because there's all kind of little molecules here that facilitate that. And there are PhDs that study the fusing of this vesicle to this membrane all their lives, right? So it's quite a bit more complicated than just what they're drawing. All right, here's another good little essay question possibly. These inter means in between, where intra, if you ever see that in Latin, that means within. So it's a little different. So in between cells, right? So like this is a cell, and this is a cell, and this is in between them. So they can connect together, basically, with these attachments. So there's three basic types of attachments, are major, right? Tight junctions, desmosomes, and gap junctions. Let's take them one at a time. Tight junctions I think of like Velcro. See the way this is a cell and that's a cell, but they're touching, right here, and that's kind of sealing it off, so water can't flow down in between these cells because these tight junctions are preventing that. Okay, so it kind of waterproofs in between the cells, or makes it harder for water to get in between them. Tends to be found near exposed, the surface of exposed tissues. Okay. Desmosomes are a lot stronger. So don't get thrown off by the name tight junction, because the desmosome is even stronger, right? So look, here's some microfilaments or whatever, or intermediate filaments rather, on inside of one cell. Here's some intermediate filaments inside of another cell, and it's almost like they're stitched together by these proteins that are going across from one cell to another. So that's really, really strong. So it's hard to rip these apart. Tight junctions, an individual tight junction is not that strong, but you got bunches of them. So yeah, that does make it, pretty tough. One, just like Velcro, the more you have hooked together, the tighter it is. But now let's look at these. This is actually, a very stable thing. So remember in the intercalated disc in the cardiac muscle? Well, lots of desmosomes in there, because you don't want those muscle cells ripping apart. So an intercalated disc is a great place to find desmosomes. It's also a great place to find gap junctions. They also call them connexins. Basically, it's just two channels lined up, because think about it, ions or whatever can pass from one cell to the other. So you can depolarize. Say this heart muscle cell is depolarizing, getting ready to contract. Well, the guy next to him, the heart muscle cell next to him, it needs to contract at the same time. So there's going to be signaling. So ions, in this case, sodium, could flow from one to the other and depolarize this cell. And then hopefully they'll contract simultaneously. So gap junctions, great place to find them is in intercalated discs. Same thing with desmosomes. There's also hemidesmosomes. We're not going into that. It's like a half a desmosome. So you can see how strong that desmosome is. I guess a hemidesmosome would be holding you down to like a basement membrane or whatever on a epithelial cell. Don't stress on it. Not going to ask you that. So gap junctions, basically two channels lined up. Organelles, we're not going into these a lot. I'll just point out which ones you need. Cytoplasm is the space, don't forget. And then cytosol is the fluid inside of a cell. All right. Not going to ask you about filament. I'm not going to ask you individual filaments like intermediate, thick, and microtubules, but just know there's a lot of stuff inside the cell, and things, it's almost like a scaffolding, and things can basically walk along, and there's a lot of little molecular motors that can bring things to and fro. It's almost like little pathways. Make sure you know the difference between cilia and microvilli. Cilia can move things along. Microvilli are for absorption and increasing the surface area of a cell. Don't forget mitochondria, powerhouse of the cell, make ATP, centrioles. You should know all of this from your Bio 130. So, yeah, I could ask a matching or whatever on rough ER, smooth ER, centrioles, nuclei, maybe lysosomes. Don't forget your plasma membrane. Yeah, I'll let you study that on your own. Mitochondrium is singular. Mitochondrion, sorry, is singular. Mitochondria is plural. See, that's why they just did RION, mitochondrion. That's singular. All right, I think we're good. Don't worry about any of that, and we'll get to this when we do more micro. But it's just showing you, say there's a blood vessel that got bent, and you cut across it, it's going to give you this configuration on your slide. Whereas if you cut here, then you're going through some cells here, and it's going to give you this configuration. You cut here, it's going to look like an oval. You cut here, you're just getting cells. It's going to look like that. So you can see how a structure is cut can change the appearance of it on a slide. And you'll get more and more adept at identifying these kind of things. And I'll show you some tricks later. All right, so that's it. Like I said, not a whole lot on chapter 2. All right, study those organelles on your own. And then I'm going to go ahead and shut this down. Okay, and I'll post it wherever it needs to be posted on Canvas and media. Okay,